We continue with the quantitative genetics subsection of the genomics, transcriptomics, and bioinformatics section. And the next talk will be by Yakov Tsepilov. It's a more specific quantitative genetics-based talks dealing with the pain phenotypes. Yakov, you have 20 minutes.
Okay, good afternoon, everyone. Thank you for introduction, Yurii. I want to talk about our study of the genetics of chronic musculoskeletal pain. Everyone knows what musculoskeletal pain is. It includes the huge variety of site-specific pains. Among the most prevalent are well-known back pain, including low back pain, and knee pain. Also, it includes chronic widespread pain or pain all over the body. Musculoskeletal pain affecting more than 30% of all adults, and in almost 50% of cases, it develops the chronic state. So it means that this condition lasts more than 3 months. Being a bit deprived of attention, it's one of the primary cause of absenteeism that costs more than 150 billion euros to the European society. According to the new version of the classification of the disease, it is classified under the chronic primary pain as a separate disease. These conditions are actually a group of mixed pain syndromes with different pathologies and pathophysiology. Given that it's very complicated to study due to its huge heterogeneity. Among its known risk factors, there are obesity, unhealthy lifestyle like smoking or alcohol consumption, social factors like depression, and genetic predisposition. It should be noted that modern dominant model of pain conceptualization is so-called biopsychosocial model. According to this model, chronic pain is a consequence of biological factors. So this is something that is broken or injured, including nervous system itself. Psychological factors like how you perceive the pain and how this perception could be affected by current psychological state. And of course, social factors like cultural background. From the genetic point of view, musculoskeletal pain is quite heritable. Heritability is moderate or rather high, up to 60% according to the twin studies. Up to now, almost all studies were mostly focused on site-specific pain separately. Quite a lot of genes were already reported, but biggest part of them goes from gene candidate studies, and only few of them were replicated or validated. In this slide, they presented the results of our last largest genome-wide association study for most prevalent site-specific pain, back pain. Although the sample size was large, close to 500,000 people, we were able to detect and replicate only 3 loci. 3 genes were prioritized, including the most associated gene SOX5 that is involved in invertible disc development. The number of found loci was relatively small compared to high. For example, if we are going to use the same sample size, we will detect more than 1,000 loci. The main reason is the huge heterogeneity of the phenotype. There are 2 ways how we can deal with it. We can increase the sample size even more, that is quite complicated and expensive, or apply new approaches that can help us to overcome this heterogeneity. So what we can use from the methodological point of view to help to solve this problem? It is known that different site-specific musculoskeletal pains share common genetic background. In other words, there is the overlap between genes associated with different pain types, but the question is, how big is this overlap? It should be noted that here and below, we will focus on 4 most prevalent site-specific pain: neck pain, back pain, knee pain, and hip pain. The question about the overlap is depicted on this slide. It is reasonable to assume that this shared, so-called shared genetic component, if it exists, by definition is something that could be considered as a general pain, something that all these site-specific pain have in common. In turn, the unique components consist all genetic mixture and heterogeneity. Obviously, if we somehow could extract this shared background between pains, we could possibly reduce the heterogeneity and increase the power of our analysis. We aim to extract this shared component to overcome the heterogeneity problem. To do so, we developed a new method that we call genetically independent phenotype analysis. The idea is simple. The measure of genetic similarity is genetic correlation. Here is the matrix of genetic correlation between 4 site-specific pain is depicted. As you can see, the matrix is pretty dense, so genetic correlations are high. We can use the canonical principal component analysis based on these matrices of the genetic correlations and obtain 4 new phenotypes, or how we call them, genetically independent phenotypes, or GIP for short. They have several properties. One of them, by definition, these components, they're genetically independent from each other. And again, by the definition, each component is a linear combination of original traits. These specific linear coefficients that called loadings. Also, the first component that explains the largest proportion of the genetic variants, we assume most probably represents the shared genetic component. We decided to apply this approach to study 4 site-specific pains using a huge UK biobank cohort that consists more than 450,000 people, mostly European ancestry. Each individual was genotyped by more than 15 million SNPs distributed across the genome. As a phenotype, we use self-reported chronic pain with site-specific chronic pain that lasts more than 3 months. The prevalence varied from 9% for hip pain to 18% for back pain. After performing GIPs for each of the pain types separately and calculation of the matrix of genetic correlations, we applied our GIP approach and obtained 4 new GIPs for genetically independent phenotypes, all these independent components. Then we discovered and replicated significant loci and performed state-of-art in silico validation techniques just to discover the biology behind these components. As a result of applying this approach, they obtained the following results. First of all, the coefficients or loadings of linear combinations. The first component looks like the sum of original pain phenotypes. The direction of, and magnitude of the coefficients for first principal component is almost the same. For other components, the picture is not so trivial. Each component except the first has the negative, strong negative impact of one of the pain phenotypes. Secondly, we estimated the genetic variants explained by each principal component for the original phenotypes. As mentioned before, the first principal component indeed explains the biggest proportion, the largest proportion of the genetic variants. And as you can see from the picture, it has substantial contribution to the genetic component of each of the pains. That is not true for other GIPs, like second component has no contribution to hip, and third and fourth component has no contribution to knee pain. Thus, we indeed can consider the first principal component, or GIP1, as something that these pains share in common. We obtained GWAS results for all principal components. For the first component, we discovered notably high SNP-based heritability, almost 30%. That is close to the lower board of heritability estimates for pain phenotypes on twins. They detected 6 significantly associated loci. 5 of them were replicated. They applied sophisticated approaches for gene prioritization in these loci. I will not go to these details because it's quite technical part of this work, but we were able to prioritize, decisively prioritize several genes. Some of them were clearly related to cartilage development and most probably related to osteoarthritis, another to nervous system development, including neuron development and synaptic transmission. To note, we have discovered and replicated only one locus for other than the first principal component. For the second GIP, we detected locus that consists GDF5 gene, that is well-known gene associated with knee osteoarthritis. Pathway and gene set enrichment analysis that we performed after revealed relatively high number of enriched pathways and gene sets, mostly related to the nervous system development, including axiogenesis and neuron differentiating. In turn, tissues and cell types tissue and cell type expression enrichment revealed several tissues related to central nervous system again, stem cells and retina. It should be noted that no significant results were obtained for other GIPs. The last but not least, we performed the analysis of genetic relations, or as I told before, the analysis of the genetic similarity, and investigated the genetic correlation of the first principal component with other available complex traits. We analyzed more than 200 different traits and diseases and identified 40 traits that were significantly genetically correlated with first principal component. Among the most highest genetic correlations were for depression, almost 60%, college completion, mother age at death, and similar. All traits form 3 clusters, 2 of them quite clear, obesity-related cluster, and mood and depression-related cluster, and one is quite large, but not so clear, but still we named this cluster as socio-demographic factors cluster because it includes all demographic traits and education, smoking, and similar. Interestingly, that the first principal component lies in this cluster. To summarize altogether, we were able to obtain 4 different genetic components of 4 prevalent musculoskeletal pains. We have detected and replicated 6 novel loci. Given the gene's genetic correlations and enriched gene sets, we suggest that the first principal component represents a biopsychological component of pain and reflects pain perception and processing. Unfortunately, nothing conclusive could be given for other components based on the obtained results. But it seems that they mostly correspond to morphology and the cartilage development and represent the unique components for other specific pain phenotypes. I would like to thank our wonderful team from several international centers from Russia, UK, and Netherlands. And these results were recently published in Communication Biology Journal. Thank you for your attention.
Thank you very much, Yakov. And we have a couple of questions so far. The first one is from Alexandra Shadrina. And she is asking, how strongly should the traits be correlated at the genetic level to be included in such an analysis?
We have, I can hear myself twice. Just a second, a technical issue. Okay, basically, we should, we applied two criteria to include traits into this analysis. One of them, that the genetic relation should be significantly significant after the multiple correction. And the second, we applied the threshold based on absolute value of the genetic relation. I do not remember correctly, but probably it was more than 30% or something. So it's some kind of arbitrary threshold.
The next question is from Alexandra Zlobin, and he is asking, is it okay to use African and Asian populations for application on study originally done on Europeans?
This approach has some limitations, obviously, but mostly in case of worst scenario, it can decrease the power of the analysis. In our case, we really wanted to include, to achieve the biggest possible sample size, to have the bigger power we have. So we decided to include non-European ancestry individuals as well.
Okay, if there are more questions, feel free to ask them. Meanwhile, so Yakov, it seems that this GIP is a rather powerful approach that can be used rather broadly, right, to decipher in the different genetic causes of, for disentangling the genetic causes of closely linked phenotypes. Are there some limitations that you can see in this approach?
It definitely has some limitations. One of them, it's basically works only if you have more than 3 closely linked phenotypes, not less than 3, I mean. So if you have only 2 phenotypes, you cannot use this approach. So 3 or more. And the genetic correlations, the magnitude of the genetic correlation between these phenotypes should be considerably high.
We have a question from Ilya Akberdin. He's asking, is it assumed that GIP1 genetic components have to comprise any gene network representing interrelations between them?
Representing what?
Interrelations between them. So basically, do the components have to be within a gene network?
So just repeat once again, because we had like some technical issues with sound right now.
Is it assumed that GIP1 genetic components have to comprise any gene network representing interrelations between them? So I guess the question is, do they have to be in a gene network, the genetic components?
Actually, honestly, I didn't get the question about which genetic networks should be comprised in the first principal component.
I'm guessing it might be about the signal picked up by this component in terms of the genes that are significant rather than the component itself.
Georgi, because of the sound issues, when you start your sentence, I hear only the last part.
Yeah, I was saying that I guess the question is about the genes that come up as significant, as opposed to the GIP1 genetic component itself. Do they have to be in a network? But that's just my interpretation.
Okay, I see. Basically, yes, they could be. This is one of the assumptions that we used for one of our gene prioritization techniques. So basically, if we found several loci for one phenotype, we really expect that these so the genes in these loci, they somehow related to the limited number of gene setting pathways. So basically, yes, we really assume it when we do the gene prioritization. And this is one of the basic assumptions that we have when we do the gene set enrichment and pathway enrichment. And
Okay.
yes, as you could see, the results for the gene set enrichment, we really have the huge number of enriched pathways and terms, and really say that it's something really specific.
Okay, we have to move on. Thank you very much. Thank you very much, Yakov. And the next will be a sponsor report by Bioline, presented by Ilse Wilman, and is 15 minutes.